All right. Good morning, everyone. I'm Charlie Munford. This is Agential Topics. Agential Topics is a meeting put on by the Agential Biology Institute, which is founded because we're interested in the idea that there may be a missing causal layer in biology, and we're building the infrastructure to study that missing causal layer. So today we have Dr. Vadim Backman from Northwestern. Hi, Stu. Welcome. And we're gonna hear from Dr. Buckman and have a discussion about the computational properties of the genome, including chromatin and his specialty in epigenetics and genetics. So without too much ado, I'll just turn it over to Vadim and off we go.
Sounds good. Thank you, and thank you for having me here. It's sounds like a really good group, and I'm kind of sad that we haven't connected. Well, years ago, would have been a very natural collaboration, I guess, or exchange of ideas. So we'll see. I might show a couple of visuals if there is interest. So I will try to be very brief and maybe focus on questions. Um, So I want to focus on something that we have been working on recently, and most of what I'm going to say has been published, and but actually some of the most exciting part, maybe, well, one of the exciting parts has not been. So as a preamble, maybe what I would say is that, and I think that probably several of us here share the same idea that the brain is not the only intelligent system in the, I'll say mammalian or complex organism, and not the only biological computational network. So the when we talk about networks, computational networks, uh, several things come to mind, and it looks like, and it almost sounds like there are a lot of networks in the body, right? Including metabolic and gene and regulatory networks, GRNs. We can probably come up with more, but there is uh, one thing that is kind of a common denominator to most of the standard networks. They have fixed topologies. They don't change, right? So if we have a gene regulatory network that tells genes what to do, which genes to express under which circumstances, it's pretty much subject of our genetics. It's not going to change over our lifespan. It can be regulated, but it's not going to change per se. So but there is at least one other network that I think is kind of unique, and that's what I wanted to talk about. It's the genome geometry network. So maybe to understand better what this is, we have to take a step back and look at the evolution of organisms, right? So starting with prokaryotes, fairly simple genome. You have pretty much every part of the gene is expressed into a protein, small genomes, relatively little regulation, and most likely, along with the genomics or gene coding, this prokaryotes developed gene regulatory networks fairly early on, right? And most of us are familiar with the genetic switches and circuits. Kind of the classical interpretation is that when one gene gets expressed, its product might work as, for example, a transcriptional factor and affect expression of other genes, either enhancing or suppressing them. And you can create all kinds of fairly complicated circuits and even some sort of fairly primitive transcriptional memory where previous exposures affect gene transcription later on. But it's purely chemical, and by definition, it cannot last very long. But for simple organisms like bacteria, that served very well. Uh, in part because their game was not to be extremely smart or adaptive at what they do. For billions of years, the strategy has been, well, population based. Accumulate mutations, random or not. I think there are different ideas about that, but the essence is uh, certain organisms, certain individuals accumulate uh, mutations that eventually propagate through the population, and the population as a whole has a better survival strategy if environment is different, if it has changed. Um, a an interesting aspect of this kind of adaptive behavior, it requires a population. It's not focused on an individual. And at some point probably came around the Cambrian explosion, something else began to happen. An organism itself, an individual, became much more valuable, and that is a critical. I think it's a critical aspect of evolution because when an individual becomes longer living, uh, when an individual becomes more important than, or at least important as a population, then you can begin to invest into all kinds of body complexities. And in fact, you have to invest in all kinds of body complexities, eventually resulting in all sorts of more complex behaviors in the brain and everything else. But the But from the evolutionary perspective, that is almost like 0 to 1 change. Why? Because you can no longer rely on simple gene mutations for an individual to survive, because by definition, that requires a progeny. It requires multi-generational adaptation. So what is the trick? Well, the trick has to be that your gene expression has to change in adaptive way in response to stimuli and or stressors. And there are really two sides of this coin. On the one hand, this can help with better survival in case of when environment changes, which it invariably does, especially if an organism lives longer. But at the same time, an added benefit, a synergistic benefit, is that you can process more information in terms of inputs and produce more outputs that may allow for higher body complexity. A liver cell in our body receives incredible number of stimuli from all sorts of other organs, which is clearly much more sophisticated behavior, has to have much more sophisticated behavior than, let's say, a cell in a hydra that is, fairly primitive. So it's the complexity, so it's adaptation on the one hand, remembering what happened in the past, adjusting transcriptional behavior, gene expression based on this history, and also being able to process more complex stimuli and produce more complex outputs. These all things come together, but they require something more complex than just gene regulatory networks that are mechanical. They don't have long memory. You cannot, and they're not adaptive. And what I think happened is the discovery of what happened in eukaryotes is the discovery of genome geometry networks. So in a complex, not every single eukaryote, in organisms with complex bodies, and this property has been what we have seen, it has been increasing as the complexity of organism was increasing as well. Genome organized in each and every cell into several thousand chromatin packing domains. Each domain is a nanoscopic structure, uh, somewhere between 50 nanometers and 200 nanometers, containing anywhere from, let's say, 10, 50,000 kilobase pairs to maybe up to megabase pair, but not more than that. So each cell has several thousand of these things. And they are all interconnected into network where each domain is a node. Some of them are cooperating, some of them are probably competing. Uh, these are not virtual networks as GRN is, or a metabolic network is. This is actual a physical network. You can see, you can touch a domain, you can physically see a node. Another important property here, what kind of the superpower of this network, is that it follows reinforcement learning. Each domain is a reinforcement learning system, and that's kind of the essence, really. So here's what happens. Uh, if a gene gets transcribed, and it gets transcribed consistently. Well, let me give you an example. For example, I'm going to expose a cell to a, well, if it's, cytotoxic stress, if it's a cancer cell, or a ischemic stress, right? Choose your pick, make your pick. Um, this ischemic stress, for example, or cytotoxic stress, is going to induce increased expression of genes responsible for example, DNA repair that might not necessarily be overexpressed at baseline. This is driven by a standard gene regulatory network by GRN. Any cell can do it. But here's kind of the magic that may happen in this more complex cell. As this gene gets expressed, it forms its own or enhances, strengthens its own chromatin domain. As a domain is physically formed to support that gene, this is actually a real physical long-term memory for the gene transcription because this gene now is poised for transcription in future times. And it gets epigenetically marked in a way that the location of this domain will survive cell division if cell does divide. Clearly not necessary for postmitotic cells, but for many other cells it is. So now we have a memory, we have created memory for the gene that if next stimulus, similar stimulus comes in, this gene is ready to go. There's price to pay because all these the domains, they compete with one another for space, for chromatin polymer. So this comes at the expense of weakening something else. So the analogy here is kind of the weights, we're changing weights in this network. We are retraining the network in real time. And that's what cells do. They keep trained and retrained all the time as we are alive. So this network, what's unique about this network, if it's morphogenic, its nodes, locations of its nodes, and their interconnection keeps changing over our lifetime. And one consequence of that, the cell all of a sudden can uh, respond to much more complex stimuli, process them, and have more complex transcriptional output. It can also be more adaptive. And we have not proven this because that would require a fairly complicated evolutionary experiment. But I think it does make sense that this kind of organism would be better positioned for a longer life, more complexity. And all of a sudden, the the tyranny of the population, so quote unquote, could be removed. So there is more incentive to invest into the survival and complexification of the organism. And that's what we've seen is that across the evolutionary tree, there's a very good correlation between how well chromatin and the genome geometry positioned and designed to create this genome geometry network and the complexity of organisms. So vertebrates in general have a very efficient system. Worms have very inefficient system. Some eukaryotes don't have it at all. And uh, across the vertebrates, they're fairly, the genomes are fairly similar in terms of genome geometry, at least how it's positioned for complexity. But there is actually progress from fishes, let's say, to synapsids. Um, so we have very well evolved genomes for this network to operate. Uh, but also there is a dark side to it, if you will. Uh, it's not a there's no answer. It's unsupervised learning. This network will continue whatever we throw at it. So if we live long enough, and that's what we've seen, it's in age cells, the cells will learn things that are otherwise not very favorable for longevity. And we have seen alterations in GGN in aging cells, clearly in cancer cells as well. This is the price kind of we're paying that at some point this network is going to learn and going to adjust itself to a state that is different from what it was when we were young. And we're currently trying to explore this for transcriptional reprogramming, things like cancer diagnostics, cancer therapeutics, etc. So let me maybe stop right here and let me ask you guys a question. Would you like to see a visual? If you do, I can share just a couple of slides for you to see how this domains look like in real cell. Yeah. Okay. So I see a thumb up. Okay. So let me share. Uh, I cannot share because I need to get permission to share.
Sorry, let me help you. Okay. You should be able to now.
Okay.
Yeah, it'd be great to see a visual. Thanks.
Yes, let's see visual thing. Okay.
Yeah, nice. Awesome.
So what you see here on the left is a part of a real nucleus of a cell. It's actually you're one of the first people who see this. Very few people saw this. This has not been, that image has not been published yet. There are just few people at NIH saw it and you guys. Uh, so this is about a little over a micron across. And each sphere on the left is an equivalent sphere, equivalent size of a Pekin domain. As you can see, some of them are, connected, some of them are not connected. So two domains on the right. This is just a part of that larger image, so you can see how they're connected. So here, the red part is denser concentration of DNA, and then there's yellow and blue is the less dense. Uh, and then if you extrapolate this to the whole nucleus, that's how the whole nucleus is going to look like. This image was obtained using a fairly unique technique that we developed called chromatin scanning transmission electron microscopy. It's not a regular microscopy. It uses a different contrast, Rutherford scattering, precisely to see the DNA density uh, correctly. Uh, so nobody has seen this before. And what you see here on the right, this is a larger portion of the cell nucleus, and then you can see that there is a domain. Let's see if I can annotate. Uh, yeah. Oops. So right here, this thing right here is one chromatin domain. You can see in the middle, there's a denser part. That is the heterochromatic transcriptionally inactive scaffold on which the what is called the ideal zone, kind of the peripheral zone of the domain sits, and that's where the gene bodies are. And the structure of this domain is fractal. It behaves as a mass fractal, which is critical because it mathematically guarantees the existence of a zone optimized at the periphery of the domain, optimized for transcription. So contrary to common or intuitive belief, I would say, when we look at dense chromatin or dense DNA as a repressive state and loose as transcriptionally active, that is actually not correct. So dense means suppression. Very loose means suppression as well. You need to be kind of in the Goldilocks zone for transcription to occur. And in part, that's what these domains create. They create ideal surfaces for transcription, but you have to have the internal scaffold, which what this domains create. So Um, they're not spherical. I mean, here what you see is just you know, it's it's shown as a sphere. It's not actually a sphere. There's a fractal-like domains. Uh, just the image that is shown here is like within a sphere. Right? So don't get confused. These are not soccer balls floating in the sun. So let's see. Yeah, maybe I'll stop here. Okay. Yeah, that's how the genome looks like. Um, and we could only really see this because we were lucky enough to have the right imaging technique.
Amazing. Awesome. Are you ready for us to ask you some questions?
I think that's a good time, yes.
Okay, fantastic. Well, yeah, so we've got plenty of time. I'd like to ask two questions to kind of refocus us on agential topics, and then we'll go on raising hands. So Dr. Shapiro has to leave at noon, so that's about 30 minutes. So I'll put him near the front if he has questions or comments. But otherwise, we'll just go by order of how people raise your hand. So at the bottom of your screen, it says react, and there's a button that says raise hand. And if you do that, that'll put you in a queue, and then I'll just call on people in order. But yeah, so just to kind of recap where we are, you described the transcriptional memory, especially in eukaryotes, that's takes place in the changes in this chromatin domain. And you talked about how that puts the importance, evolutionary importance, on the individual as opposed to the population. And so, as genes get expressed, it changes the weights in the gene regulatory networks, and that's a form of learning at the cellular level.
Hmm.
So just to like blow that out to the bigger scale, the question, it's going to take me a second to introduce this question, but um, so there was a moment, I'm sure a lot of us have watched the debate that happened between Richard Dawkins and Dennis Noble. And Dennis has joined us in this group several times. They had a debate a couple of years ago at the Arts and Ideas Festival, and Richard Dawkins made the claim that you could take Dennis Noble's genome sequence and etch it in stone, and then 1,000 years from now, you could recreate Dennis Noble. And Dennis objected to this claim, and he said, "Well, where would you get my mother's egg cell as it was in 1939 or whatever it was?" And and so I think that interchange expressed for me two different, very different ideas of what kind of computational system um, the zygotic cell is. And so, on the one hand, the sort of received idea that most people outside of this group might might take for granted is that the organism is a sort of digital computer.
Hmm.
The the genome is a digital code that's edited by random mutation and natural selection. The input is the environment, the output is a phenotype, and what the computer is generating is a an algorithm that computes evolutionary fitness. Okay. But a lot of us in this group have this sort of more read-write dynamics in our minds and learning in our minds. And so some of us think of it as a different sort of computer. So an analog computer, where the code is all the relationships in the cell and also the cell's relationship to other cells. And the output is also those relations. And the input's still the environment, but it's computing something about the environment that's different from just a pure readout of evolutionary fitness, computing something cognitive about the environment. So, my question for you, Vadim, is are those two models in tension in your mind? Are they compatible, incompatible? Is there an option C? How does that frame your work?
That is a great question. And I'm trying to think how to answer this question without spending like a couple of hours answering it. Uh, so Hawkins obviously is a giant, and beautiful mind. A lot of things that he said is, profound and has changed biology as we know. I do have to say that, personally, a lot of, I'm not sure I would agree based on what I've seen, based on the data that I have seen, uh, particularly our data. I don't think I would agree with the selfish gene idea in general as the premise. Um, that's not what we have seen. What we have seen is the importance of computations in biology in general for organisms. And in particular, what we've been looking at, the genome itself as a machine, physical computational machine, uh, where the computational complexity has been steadily marching forward, at least over the past several hundred million years, uh, focused on better cooperation in among cells, let's say, right? More processing more complex inputs, producing more complex outputs, having longer term memory within and across lifespans. These are not features to me that point to the selfish gene that is just trying to propagate. This points to me to the idea that computations is, or ability to compute for a living organism is paramount. And we all know that having more nodes or more ways to compute, well, results in better computations. So that's at least what I think I have seen in our data when we look at the genome. Now, one thing that you know, I can say, you ask a question whether, you know, these two paradigms are mutually exclusive. I don't think so. So going back a little bit to facts here, what we know right now is that the potential positions of these domains, they are indeed genetically determined. So I'm born with certain bookmarks, so to speak. And we know there are very specific sequences that put bookmarks in between where my cells can form domains. I'm born with that. I cannot change that. That is a slow boiling mutational evolution. However, whether a domain is going to be realized in that place, that's a different story. There is no genetic code for it. The only way this is decided is through the experiences that a cell in my body is going to make over its lifetime, and or lifetime of its progeny, etc. Right? And also uh, epigenetic modifications that my first cell, right, the fertilized egg, uh, acquired. And that goes back to to my parents, let's say, et cetera, et cetera. So we know that some domains can be very short-lived. They can have a lifespan of like a few hours, very short-lived. But some, and many, can live for decades. And I don't know what the expiration date is because some of them are extremely long-lived, right? So there's a whole range of memories here. And so yes, the locations are genetically determined. But you can realize a domain or not. You can also combine them together, or you can break them apart. So you imagine kind of reading so many possibilities for a reading frame. Number of combinations that are not genetically determined is literally astronomical. So I think that probably if you want me to say that the truth somewhere in there's a way to reconcile that. I think there is, but it's I think there's certainly more to it than my genetic code.
Totally. Um, cool. Well, my second question is about, so I'm particularly interested in the sort of contradictions, wherever we can find contradictions between learning and evolutionary fitness. And So one of those contradictions I think about a lot is senescence. So like we have this, we have this something that appears to be a program that our cells learn that doesn't result in us exactly being a Kantian whole. We actually are so Kantian whole for a while, and then we sort of self-destruct, and it's hard to distinguish the changes that help us develop from the changes that end up destroying us. And so so I wonder if this um, if your work and your investigations have reframed cognition at all, I mean, senescence at all for you, and how you think about distinguishing those processes from one another.
Senescence is a very interesting process, and frankly, I don't have all the answers. We have been looking at senescence too. So so here's what we know. There are all shades of gray, of course. But if we want to put things in terms of GGN, if we want to put them in like baskets or two by two matrix, right? You can't do anything without two by two matrix, right? So on the one hand, you have differentiated cells, especially post-mitotic cells. They have a fairly settled, very mature GGN. So most domains are large, mature, very, very stable. It's very difficult to create new ones. It's kind of conservative deep attractor system. Uh, in another corner, you have cancer cells. And what we have seen in cancer cells is almost like the opposite. Uh, they're like bubble with domains. Their GGN is has fast turnover. Domains appear, new memory appears, gets degraded, new one appears, very, very plastic. Senescent cells, which which we again I don't fully understand what what they are from this perspective and why we have this mechanism, but they do have a fairly interesting third corner or third state of GGN or third extreme state, if you will. Um, they look like mashed potatoes in terms of GGN. It's like um, the so new domains, which mean again, it's kind of new memory, can form fairly quickly, but they never really develop into any. They don't mature. It's like they like almost like a flicker of memory shows up and then it just dissipates. Nothing gets seriously done. Nothing gets seriously, so these nodes don't seriously get created, basically. Why? So that we know about the structure and what it does. What I don't know, I can only philosophize or speculate at this point why the state actually exists. And one answer that, one simple-minded answer might be there is no rhyme or reason for it. It just happens. Why? Because again, GGN is not there is no, right or wrong answer for GGN. It's not, it's unsupervised learning. So if you expose a cell to, let's say, harmful stimuli long enough, then it might end up in one of two states. One of them may be this hyper uh, morphic, if you will, state in a cancer cell, and another one is a mashed potato senescent state. It just a byproduct of cell being exposed to wrong stimuli for a long time. I don't know why we have it.
Awesome. Yeah, great. So we have a ton of questions. Guys, I'm gonna give Dr. Shapiro has a time constraint. So, Dr. Shapiro, if you have any questions or comments at this point, I'll give you the floor if you have anything. Otherwise, I'll go down the list.
Well, the one question that I have is what is the sequence composition of the different parts of these structures? And does that have any influence? I'm sure it must have some influence on the way the genome folds.
Absolutely, it's a great question. So first of all, there are, as I mentioned, there are very specific kind of bookmarks, quote unquote, but what is called hinges. So if you have a domain and they have another domain next to it, let's say there is a spacer in between that helps them potentially stay separated. They don't have to be separated. They can merge. But if the system is such that domain A and domain B, they are independently activated, they will stay separately. And this hinge or bookmark is what helps them to be separated. They have fairly specific structure. So if um, in in the language, let's say of molecular biology, they have uh, uh, CTCF anchors. They have a anchor for a transcriptional site. So they have very fairly specific sequences scattered throughout our genome. They create positions for domains. They do get mutated in diseases such as cancer. In aging, they frequently get mutated as well. So there is there's a regulation of this hinges or bookmarks that happens as well. One interesting thing is that uh, they don't their structure can change from individual to individual or species to species, and their location on the genome can change. And that's actually where the story gets more interesting. And I don't fully understand all the significance of it, but here's one fun fact. When we compare, we frequently compare, let's say, us with chimps, right? Or apes in general, right? Uh, and uh, and we all heard number like 98% similar genome, right? Or that we're 55% similar. We're 55% banana, right? 98% chimp, and only 2% or less than 1%, right? Our genomes are different between chimps and us. Uh, so this difference there might be some very, very important couple of genes, right? But here's the interesting thing. If we look at the uh, mutations in these hinges themselves that determine separateness of domains or where they are on the genome, then the difference increases anywhere from 8% to 20% between chimps and Homo sapiens. So it almost looks like, yes, our genomes overall are very similar, but it's not about the proteins. Our proteins are indeed very similar, but which proteins are made under which circumstances, that is actually very different because 10 to 20% chance, 10 to 20% differences, that is actually huge. So we are very different from this perspective, from what our cells do with our genes, that is very different. And the same holds for other related species, like mammoths and elephants, right? So makes me wonder what kind of gray wolves, how to make gray wolves into dire wolves, right? What exactly we're making there, but it's a different story. But we're certainly very different from other primates in that aspect.
Thank you.
Awesome. Mary,
You
you,
might be muted.
you're still muted, Mary.
Sorry.
There we go.
This is a fascinating, still muted. I'm very interested in the uh, what constitutes a holon and whether or not the invariants, how far down they go, how far down holons go, and it struck me that you've been describing what I see as some of the invariants, and I'm wondering if I could ask you if these nodes if they have anything that you would consider an energy expenditure within them that they manage to build themselves, and if you consider the nodes themselves to be self-regenerating. And the answer to those questions would be really amazing because that would indicate something about how far down holonicity might go in the cell.
And that's a great question, and we're just beginning to understand some aspects of it. From the energy perspective, maintaining GGN in general and creating domains, especially creating domains, is an extremely so that is It is enzymatically driven process.
Mhm.
It is a physical process, so it naturally happens in a polymer, but domains don't form sp spontaneously. It's not like a phase separation thing where just things aggregate on their own. It's enzymatic self-assembly based on very strict rules of physics. And in order to create a domain, the first step is to create what is called forced returns, to move chromatin against its equilibrium, entropically driven state. And this can be done by two ways: by active transcription, creating transcriptional loops, or by active loop extrusion, both processes being very energetically consuming, of course.
Yes.
But then the second thing, that's only part of the problem. But then you need to mature the domain. And that happens by deposition of epigenetic marks guided by diffusion gradient. So the guidance is natural, right? It's like it doesn't cost you anything. Uh,
Yeah.
but deposition, chemical deposition itself, again, is energetically consuming. So once you create a domain, then it becomes much less energetically consuming to maintain. But creating a new one is a double whammy because first it requires, again, new energy consumption. And second, this new domain is going to compete for two things: A, space, and second, the polymer itself. So the polymer, availability of polymer is a currency here. So that's why some of these nodes can be quite stable. It's the newcomers that have to really fight for it.
But the nodes themselves aren't actually doing the maintaining. It's other processes within the cell. It's
Yes.
the other transcriptional processes. Okay,
Yes.
thank you.
Cool. Henry?
Oh, hi. I really enjoy it because almost 25 years ago, I studied chromatin domain, how regulate the gene regulation. And at that time, we are actually much resolution is much lower, but the conclusion is the same because we actually thinking we want to actually compare different species, compare normal and cancer, and you actually get more detail. So I think we can discuss more. But one of
Let me just say that absolutely, and I've been a big fan of your work too. What's interesting, just personal anecdote. So I've been a faculty for 25 years, and in year 2000 or 2001, I went to one university and was giving a talk, like applying for the assistant professor position, and I was talking about chromatin structure as a potential regulator and potentially domains as a regulator of transcription. That might be important in cancer, would be interesting thing to study. And that is why I did not get the position because people said, well, that's just nuts. We all know that it's all about genes, right? What the heck is he talking about?
yeah. So that's exactly. So I'm so glad. But then I feel that our work actually we have our own gap. You have your gap. Actually, it can very nicely match this gap together. So I will quickly just give an outline a little bit. So recent years, we actually focus on the concept of the division of the information
Hmm.
because we found that the information actually at a different species level is completely different from the regulation at the tissue level of the same species. So some of the phenomena actually answer this question because we know the temporary formed functional domain that is most of the related to epigenetic regulation. But
Hmm.
from evolutionary scale, if you compare with, I mean, I'm glad you have the data of chimpanzee and the human because we actually thinking they have totally at karyotyping level different coding. Therefore, they should have massively different in terms of compare with the gene contact. Because we
Hmm.
thinking the gene, the physical position on the chromosome, and in the interface chromatin domain, that physical relationship is a coding
Hmm.
system. So if you change that, same gene do different thing, right? So this is one of the idea. So in that sense, now we with the idea of division of the information, we really need to separate to compare with different species For example, the cancer, you said there's totally there's no structure. Yes, the cancer is a super system that don't have inheritance to be fixed. That's why all of these dynamics constantly change. Now we actually write a paper thinking cancer is a new form of biological entity, right? So that's quite interesting. Maybe we should
Hmm.
cite some of your data to support the
I
idea.
absolutely, I would, I would love to talk more, and I think your ideas about genome chaos are also very relevant and very powerful. Uh, so I would love to explore this more, and maybe we can, yeah, it would be good to contribute to your upcoming paper.
So now the quite interesting is use your system. We actually can study the mismatch between information, between carrier, and between ecosystem,
Hmm.
because most of the information did not coded yet. Because we
Hmm.
always thinking is coded, but actually biological system is coding the process. And
Yes,
the products is environment interact with the process. They have nothing to be coded.
exactly.
So that is a
Yeah. Yeah.
misconcept. We actually need to really push this one. So how, because we actually found that many of the information is during the process of developmental process, during the aging process, that process themselves constantly create new information which beyond the genome.
Hmm.
However, the boundary is defined by the genome. So no matter how new they are, they're still with that particular system. So that's why the genome chaos is actually break up this boundary. That's why
Hmm.
the power is from there, because they completely form the new information like the cancer. Then they never have this linearity from the previous cancer. They're from a normal cell with complexity, but then the emergent cancer. So this discontinuum of information flow, and then they actually become so powerful in the future. We're worried about it. They will take over the world because of that,
Hmm.
right?
Right.
So that is very important. So I just then I one thing is just I need to stop because too many stuff needed to discuss. But the genome themselves is very important because they define the boundary of this all this for where to go, where not to go. But the where to go, where not to go only fulfill the developmental or environment adaptation sense, but they should not change the evolutionary sense in terms of species.
Hmm.
So separate those ones, so your technology really can bring some of new insight to this field. So I will stop here. When we have time, I can ask a few more. Yeah, thanks.
And it would be great to connect offline. I would love to talk more.
Awesome. Great. Kate?
Me?
That's you.
Cool. Oh my
Sorry.
gosh. Vadim, I'm thrilled to hear about your work. I've been walking backwards from psychology and studying emotion for a long time, and I was hoping that over 10 years ago, we would shift away from the selfish gene scenario to the self-regulating genome. And I remember mentioning that in a talk, but lately I'm actually thinking it's actually a self-regulating organism. And the genome is but one of many beautiful layers within the system, very much like a transformer network, if you will,
Hmm.
computationally. So one of the things that I was really fascinated by in my study of sensory motor systems was that there is in the bacterium, there's a real-time sensory motor control where that can change the flagella to go counterclockwise or clockwise. It basically moves the bug forward or tumbles back. And that is a real-time mechanism that's due to a phosphorylation that's in direct response to the gradients in the environment. But the cool thing is there's this, and in the same time, there's a methylation mark going down that category that provides a little memory. It provides like a short-term memory that lasts for several hours about where it went, whether it went
Hmm.
forward towards something or whether it tumbled away. So it sets up a memory, which is um, essentially the kind of learning that you're talking about, Pavlovian learning in a bacterium. So my question to you is, what you're saying is absolutely beautiful, and it sounds like it's connected to the sensory motor regulatory chemistry, because methylation is offering a memory, but it's on the receptors, the cell
Mhm.
bodies of the cell receptors, but that memory, it sounds like there's a higher level going on here with your geometry that's connecting higher level still to Mike Levin's bioelectric patterning. So that's my question. And I would love to talk to you myself as well. I'm assuming if I just Google around, I can find your work.
Yes, for sure. So the network images that I showed, they have not been published yet. We are working on a paper, so that you will not find. But the work on domains, yes, you will find that. And yes, I would love to talk offline as well. So there are a couple of things. First of all, my short answer is I think you're spot on, and hit the nail on the head in everything you said. The uh, there's clearly very important contribution from electromagnetic or electrostatic, electrodynamic effects as well. That's actually another paper that we published on this, but another full paper is going to come out in the next few months in, I think it's Advanced Science, if I remember correctly, where this explored in a little more detail. So ions play, nuclear ions in particular, play a very important role in creating domains and second, maintaining them. And of course, whenever we think about ions in general, things like magnesium, calcium, especially magnesium. Um, and this is in part important, by the way, because DNA is by definition a highly negatively charged molecule. It's actually an electrical monster. So the electric potential, literally electric potential of DNA, naked DNA, is within a few nanometers of it, is several gigavolts. And so if if any of us is not familiar with gigavolts is, let me put it this way. If you expose me to even a fraction of that, I'm going to explode, literally. I mean, like I'm going to disintegrate. Uh, right, like in a sci-fi movie.
Yeah.
So that's how anomalous DNA is. So in order for any domain to be formed, it has to be screened. And this is accomplished in part by divalent ions such as magnesium playing the central role, also sodium, potassium, but magnesium is the central one. And that's why it's one of the reasons why ions are so critical in maintaining the structure, allowing the structure to occur. Uh, and their magnesium or other ion concentration in the nucleus, of course, is in part regulated by electrical currents that we have. Uh, there is much more to these processes, and they're really fascinating processes. So clearly, electrical currents, whether AC or DC ions, this is all very, very critical aspects. And there is also a direct connection to the position of, let's say, methylation marks. So uh, so methylation marks are not all of them are repressive, but many, probably most are. So some of the most frequently found are H3 K27 trimethyl or H3 K9 trimethyl. And here is the kind of an interesting part is that these marks accumulate, a lot of them accumulate in the course of domains. So which
Cool.
may seem like almost like counter, I guess, intuitive, because we associate repressive marks with gene suppression, right? But
Right.
what
Right.
turns out to be the case is that you need both. You need a coherent, you need a combination of repressive marks, such as repressive methyl pro-transcriptional marks working together in the right geometrical pattern for
Yeah.
transcription to occur. It's almost
Yeah.
like reminds me, I have a 13-year-old, so you ask me about a Disney movie, and I'm an expert, I can quote it. Like Inside Out, you need to have sadness and joy at the same time. That's the same thing. You and And a lot of us have seen that. Those who have studied genomic processes, transcriptional process, we have seen that if we get rid of a lot of methylation, transcription actually is going to go down, which
Yep.
makes very little sense, right? But it has to be in the right geometrical pattern. So this repressive, let's say, methylation marks in the core of a domain, they maintain the integrity of a domain, which allows your chromatic periphery to become an ideal place for transcription to occur. You remove that, and you remove the Goldilocks uh, environment for gene bodies, especially for exons. And and an interesting caveat here is that this deposition appears to be, at least in part, geometrically guided. So a few years ago, and it wasn't us who figured this out, uh, but it was found that most components of epigenetic modifiers, epigenetic enzymes that deposit methylation marks, especially, let's say, H3K9, trimethyl, et cetera, they are very small compared to euchromatic enzymes. So they can actually penetrate by the nature of diffusion. They can penetrate. They're naturally enzymatic and not non-enzymatically, but entropically guided to the core of a domain where they begin to dominate, overcompete everybody else, and deposit more methylation marks and vice versa. So geometry in this case is one of the physical guides for where these marks accumulate, which actually is a really beautiful system if you think about it, because these enzymes, they're not loci specific. They don't know where to go. So genomically, the program there where they can go would be an extremely computationally, or from gene perspective, task would be very complex to program all of that. So what you do is you create a rule, a physical rule that uh, naturally takes them to the right place, which is actually a very elegant solution. So these things, they feed on each other. And of course, as soon as you deposit the marks then in the right places, then you mark your geometric structure. And if cell divides, then it remembers where to put this domain again. It doesn't have to reinvent itself. It probably still needs a little bit of learning, which is probably why most cells, after they divide, they don't divide into exact replica of itself. They undergo several days of further differentiation most of the time. But still, the blueprint is there. So it can be it's a really I think it's a really beautiful system from the engineering perspective.
Can you tell me what the physical rule is?
Physical rule for deposition.
Yeah, I mean, you're basically attraction, repulsion, symmetry,
Right. So
gauge symmetry.
there are actually several. There are several physical rules here. One
Okay.
of them is what happens in the ideal zone, the Goldilocks zone, is that there are two things happening physically. One of them is a transcriptional reaction is it requires two things to happen. You have to deliver transcriptional reactants, which is accelerated by diffusion. So diffusion coefficient, diffusion rate is important here. The second thing is it's the binding of the complexes, transcriptional complexes that have to happen. And they have the opposite, uh, they're opposite functions of local chromatin density. So diffusion gets killed by higher density. But because of the excluded volume effects, formation of complexes, and most enzymes are complex, they are facilitated by higher density. So as a result, because you have two opposing reactions, effects of the density, there is a kind of ideal and optimal position happening at about 35% of volume occupied by chromatin, where diffusion is not too decreased, but then formation of complexes is enhanced. And this is the place where most transcriptional complexes can happen. You try to do the same thing in a very loose, let's say, environment, And then your transcription complexes are just not going to bind. They're going to form very well. They will attach and they just diffuse away. Just they're not going to stick around. That's one thing. Another thing is the dimensionality reduction in the search, in transcriptional search, which makes transcriptional search much more efficient when domain exists. Another physical rule is that, as I mentioned, most heterochromatic enzymes are several times smaller. They're about 2 nanometers on average in size, where euchromatic enzymes are about 6 nanometers in size. That translates into several times better diffusion of heterochromatic enzymes in dense environment, which means that when they get preferentially accumulate, just because you can keep diffusing into denser core as domain is being formed by transcription forming forced return. So they get inside the domain and they can stay there and they can deposit all these marks. Euchromatic enzymes just cannot get there, which means heterochromatic enzymes have no competition. And so this differential diffusion, what modeling results show and validated by experiments, is that results in preferential accumulation of uh, methylation methylation depositing enzymes in domain cores and vice versa. So it's just a geometrically or physically guided system without uh, programming it genetically.
I love it.
That's so awesome. Gather, I'm so glad you asked that question and you followed
And
up
and,
on
and,
it.
and by the
That's
way,
so
so it's actually what I really, as an engineer, I'm I really think it's a really smart system also because you create rules. So the geometry is three-dimensional, but you cannot pass three-dimensional geometry to your offspring, right? Because we have to replicate the DNA. We have to turn this three-dimensional structure in one dimension. In one-dimensional structure, how do you pass a structure? You can't, right? Unless you mark the locations, you make a projection from 3D to 1D, you mark the locations of where these domains need to be rebuilt, which is what these epigenetic marks provide. And then when you replicate the cell and the cell divides, replicate it and the cell divides, then you can begin to rebuild the structure again because you have this one-dimensional blueprint. So you can pass one-dimensional information, you cannot pass three-dimensional, but you can pass three-dimensional information if you project it on 1D.
Awesome.
so
Uh,
fascinating.
Can I
Awesome.
say one quick thing? My husband Stuart and I have been arguing over whether Platonic forms that are more stable and eternal are important to identity and agency, or Aristotelian forms, which are dynamic and ever changing. And you just described how it can be both, which I've been arguing for. So thank you so very much.
Very interesting, very interesting. I would love to be a fly on the wall and to listen to your debates.
We'll do a Zoom, the three of us.
They're they're a pretty fascinating couple. Great. Um, welcome, Giovanna.
Hi, thank you so much. And thank you for this inspiring talk. I have a bunch of questions, but I guess the one that I'll ask concerns precisely this, the role of the genome in morphogenesis. So, if we take Levin's idea seriously that an organism already knows its target morphology, so it in a way needs to have a memory, or that cell needs to have a memory of the organization that it was a part of. And where is that memory stored? Does it exist anywhere as physical blueprint, or does it exist as an absence that is just sensed by the organism? But you do from your narrative, I do think that genes might play a big role, not in a sense that they are bottom-up constraint from which morphogenesis unfolds, but that like because if we think of Levin's Picasso frogs that self-correct during development, so it can't just be like a mechanical bottom-up thing. But so my question is whether at the time of self-fertilization and then differentiation, whether that parent cell has sort of epigenetic or the genetic expression that serves as sort of a mnemonics from which the organism kind of, remembers or reconstructs the memory of itself, right? And then once it has an idea of, okay, this is who I am, then during differentiation, even in if perturbations happen, it can kind of revert back to where it's supposed to go. So yeah, I don't know if that's a clear or
That is a great question, and uh, I clearly don't have all the answers. But here is one thing maybe I want to propose, potentially to speculate and think about. Uh, um, so here's what we know. There are some things we do know, and what we do know is that when the egg is fertilized, it's not a blank slate, right? So there are specific uh, methylation, acetylation marks, and I'm not talking about DNA methylation per se, but specific histone marks that mark where domains might or might not be. So this information is already in the very first cell when we begin to develop, which is why I I would probably disagree with the argument that it's all about our genes and as a blank slate. No, I don't look at myself as a fully closed system, right? Not only I communicate with a lot of other organisms and stimuli, but I am not just what I am after birth, but I was influenced by a lot what happened before me and my parents, et cetera, et cetera. But so we know it's not a blank slate. So what we also know is that when we look at, let's say, embryonic stem cells, they're kind of weird things. I mean, I mentioned several extreme examples, cancer, senescence, differentiated cell. Well, in another quarter, we have the embryonic stem cell. It has some domains, but they're not fully formed yet. They have a potential. Um, and then as the cell begins to differentiate, uh, different domains begin to form in specific places. For example, if we take let's say, iPSC cell, and begin to differentiate it into a muscle, one of the critical genes, and muscle is a good system because there are fairly few genes that are needed to be expressed for muscle to stay in a muscle state. And one of them is a myote gene. And what we saw is that as the cell differentiates, a domain right around myote begins to form. It wasn't there in the beginning, but it begins to form. How does the cell know how to do it? And again, well, the answer is I don't really know. One thing that maybe I would like us to think about, maybe the rule is not in some blueprint that exists. Maybe the rule is the rule of inference. Uh, and I think a lot of people in genetics, they like to think, or epigenetics too, they like to think in terms of there has to be a blueprint. Because we keep thinking about the right genes, wrong genes, gene mutation, right? If we fix gene mutation, we're going to be okay. DNA is a blueprint. There's a right or wrong version of it, right? But and the work, for example, by David Sinclair at Harvard, they've been trying to do rejuvenation based on taking epigenetic states back to the youthful state where they should be. And maybe, just maybe, there is no such perfect blueprint. And from this perspective, the only thing that we have is the rule really is or the blueprint is the learning process itself. So if I'm a muscle cell and I'm beginning to be exposed, just like in reinforcement learning process, I'm beginning to be exposed to certain chemical or mechanical stimuli around me, that is going to drive preferential formation of certain domains that will keep reinforcing. The system will keep reinforcing itself to the point that a new domain around myote gene is going to be developed. And so that is going to be my direction of differentiation. And then every cell will have the same thing. All cells are quickly learning and developing their domains depending on the environment, which translates into enhancement of GRN and how which genes get expressed eventually into consistent differentiation in organ formation. Uh, Again, what I just said, it's not a fact. We haven't really investigated this thoroughly, but it's just one possibility.
Awesome. Great. Anthony?
Thanks, Vadim. Fascinating talk and tantalizing slides. I wasn't able to synthesize all of the information on them quickly enough. But I'm inspired in this question by Robert Rosen's concept of an anticipatory system, one that contains a model of itself and a model of its world. But most important for me in the context of the question is this concept of temporal spanning, that a system to anticipate has to have a fast clock that describes a future state that then informs a slower clock how to react to that future state. I'm also thinking about the papers that have been written on the genome as fractal antenna, and I'm interested in information encoding on topological manifolds, and that's where you are. Thinking about the chromatin as a spatial encoding mechanism, but I'm very interested in your thoughts on the transposition into the frequency domain or the time domain, and the concept of harmonic synchronization across the scales. Do you think about that in the context of your work?
I do. We haven't really explored this or published this, but you're absolutely right. It's something that we've been discussing a lot. I think it's a really good question. It's a really good direction to think about.
Fantastic.
And also, what you mentioned, I think you're essentially referring to out of distribution computations.
Yeah.
Uh,
Asynchrony as a computational signal. That's what I'm thinking
right,
about. Yeah.
right, yes.
Mm.
These are very good questions. And another personal anecdote that Don Coffey, I don't know if you, uh, okay, so
Not yet.
Donald Coffey from Johns Hopkins. Um, back in like 90s, he was talking about very similar questions, including synchrony. And he passed some time ago. Was a brilliant, brilliant, brilliant man, and everybody thought he was a little. Well, first of all, he looked like one of the founding fathers, and like Ben Franklin. He really looked like Ben Franklin. But then he was a brilliant surgeon. But then when he talked about synchrony and related aspects, a lot of people thought he was just smoking something. And
I know that feeling,
I
by the
go
way.
back, I go back to these ideas frequently, and I think it would be fascinating to explore them in the context of the genome as an intelligent system.
Thanks. Great.
I'm
Awesome.
Yeah, there's a really fascinating considerations.
Awesome. Joanna, you still there?
Hi. Yes, I am. I'm not in front of a screen or a camera, but I can hear you. You can hear me?
Yep.
Yeah, we can hear you.
Hi, Vadim. Thank you so much for your talk. I did see the videos in the beginning. It was stunning. Thank you so much for sharing it with us. It's always a delight to be with this group. Um, and also shout out for engineers coming to science because I always think they bring a lot of pragmatism. So I was wondering, a very simple question. Do you know if bacteria have anything like this? Because I think the work you've done was in eukaryotes, which we know have a lot more complexity in their nucleus. Then I had a second question about DNA can last for much more than 2 decades. I mean, at least the sequence, we know that we have sequences that are 2 million old. So I was wondering if this type of 3D complexity could also not only withstand time, but facilitate the withstanding of time of DNA. So if the 3D complexity genetically can be much more fundamental than we are thinking is my question overall. Thank you. Thank you so much.
Well, thank you for your question. And you were breaking in one place. It might be my connection, but I think I got the question. So I don't know if bacteria have a similar system. I would not be surprised if they have something. If I mean, clearly not as complex or as not as efficient, but some sort of system that would mimic it in some way. I wouldn't be surprised if it's something we would love to look at. What we do know is that um, in order for this domain network to be very efficient, these domains need to have a fractal structure. And again, mathematically, why it's important is because is the fractality of the structure that mathematically ensures the creation of this ideal memory zones for genes. So now, if you imagine now that we have, surface, well, proverbial surfaces, these are not real surfaces, right? Like fractal surfaces, but imagine the surface and there's a volume. You have to have the right ratio of the material in the volume versus the surface for it to be stable. Otherwise, if you have too much in the volume, too little on the surface, or vice versa, it's not going to be a stable system. Um, and we can calculate that. But the question is, what do you want to put in this volume? Because the volume is going to be transcriptionally suppressed. Well, you can put a gene in there. In this case, it's probably not going to be. It's going to be extremely difficult for this gene to ever be expressed, especially if it's a mature domain. Is it possible? It's possible. It's probably not the most efficient way to do it, but it's possible. A better way to do it, a smarter way to do it, is to have enough junk DNA, quote unquote. By the way, and I don't like this word junk because I don't think it's junk, but let's call it junk. DNA that does not transcribe proteins and package that thing inside the interior of a domain. Things like introns or extragenic sequences. We can put them as scaffolds and let's say exons would be on the surface. And what we have seen is that in organisms like vertebrates, for example, that's exactly what happens. And we by looking at gene sequences, we can figure out how optimized the genome structure is to create this efficient system. And in mammals and birds, the answer is we're very well optimized for that. For most genes, there is a right amount of surrounding non-coding sequences to pack the right volume. And it's not a coincidence, again, because there has to be the right ratio of the two. So our genome's involved for that. If we take a clock back and start looking, let's say, at flatworms, for example, much less efficient configuration. Go back to, let's say, yeast or other simple eukaryotes. It's a very much a linear genome. You cannot create volumes anymore. Now, it does not mean that you cannot create domains. You still can, but you have to sacrifice some other protein transcribing genes to do it the right way, and also risk that it's not going to be such a stable system. So bacteria would also, so even within eukaryotes, not all eukaryotes have that. The more complex body is, and it's not just about multicellularity, it's really about complexity of the body. The more complex body forms organisms have, the more likely their genome is going to be optimized for this kind of packaging. So bacteria don't have that, and they don't even have histone structure that eukaryotes have. However, it does not mean that they don't have a somewhat similar, at least in some shape or form, similar system to that. It might exist, but there would be one thing is for sure there would have to be compromises, even if it does exist. You would have to sacrifice some genes as scaffolds, which would probably make it a little less versatile.
But with your technique that you smartly invented in your lab, could you look
We
in the
we
same
can
way,
look.
right? You can look,
Yes,
you could just look.
we
Yeah.
can look at it. One yes, and we are looking at this. One organism, absolutely correct. Yeah, we can look at it. One organism that or group of organisms that I think can be particularly interesting for us, and we've been studying it, is coral reefs, corals.
Oh,
Why?
interesting.
Yes, well,
I
because
was sorry.
tell me,
I
yeah.
just wanted to tell you before I forget that it might be also interesting, this technique, this microscopy, to look at bacteria in biofilms because they
Hmm.
do segregate a lot of extracellular DNA. And I'm pretty sure people have no idea about the impact that these molecules have outside the cells structurally in the geometry of the assembly of the biofilm itself. So I don't know. It just seems that your technique has so many cool applications. So congratulations.
Well, thank you. That's a good, yeah, you're right. I mean, I actually, that's a gr brilliant suggestion. Yeah, I haven't thought about biofilms. Very good. Thank you so much for suggesting that.
Thank you. Thank you.
Fascinating. That's totally fascinating. So Mary, you're up again. Mary, can I ask you to before you ask your question, can you provide a little bit more context on your concept of stacked holons? Because those of us who know you well are pretty confident that it's pretty deep and fascinating, and we haven't really heard it. So if you could you give like a just a 2 minute update on your thought process about the holons?
Sure. So I've been looking at what are the invariants of levels of cooperative life. Life is cooperative at every level, from the endosymbiont up to an ecosystem, let's say. And there appear to be invariants to the holons that are formed. And we're actually testing this in software now, and it's really fascinating the way that it is demonstrating a relationship that doesn't even have to have a physical structure. And what I'm seeing over and over again is that I can predict what's going to be found at different levels by looking at the holon invariants. One of them, for example, it was related to my question. I just put it in the chat. There seems to be a lamellar boundary, a multilamellar boundary on holonic structures. So if I can ask a question and it turns out that lo and behold, there's a lamellar boundary, and in this case, it's indeed the lamellar boundary that we've identified as the memory structure for that we're speculating is the memory structure of a cell, and that is the multilamellar membrane, and it is in fact tethered to these nodes. So my question is, have you looked at memory as a possible placement for that? If you want me to go through all nine of the invariants, I could, but maybe we could set a time to have a chat about that because everyone in this chat has asked questions about the holons, and I'm just sitting here going, "That's number nine. That's invariant eight." In sort of keeping my holding my tongue. So sorry, Vadim, I didn't mean to jump in on you in your on your lecture.
No, 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 very good. And if I understand correctly, so holon is a is a I guess an I guess an entity, right? That is a
Yeah.
whole in its own right and part of a larger
Yes.
system.
And
I
appear to be invariants. A bounded cavity that is resonant
Yeah.
either phonically or photically. Memory, leaks and lacks. That's an idea that I developed out of Terence Deacon's presentation to us. It appears to have circuitry and circulation.
Hmm.
Um, and these invariants repeat over and over. And every time you look at one, even the sun has a lot of these. It may be a protoholon or a pseudoholon, but it's really
Hmm.
fascinating to when you see that each level has them. And so I wanted to ask about memory because if the more I ask the questions, the more it looks like your nodes are holons. And if there is indeed memory at a membrane, that means that not only is a holon something that is relational and not tied to physical structure, which my team is theorizing, but it's also something that goes down below the the a primitive bacteria and smaller than a the parts of the cell. So
So let me clarify. So are you thinking about that the genome geometry network is you know one of the so
it's
scale
holonic.
of
It
the
looks like it might be a holonic level.
colonic level, right? Yeah.
It looks like it might be a holonic level that's smaller than the mitochondria in the other endosymbiotic levels. We don't know, so it's really fascinating. I'm happy to send you the invariants, and we can bang on it together.
I think that would be very interesting. So
Yeah,
before this meeting, I had not thought about GGN as a whole on, but I think it actually makes a lot of sense. And I probably wouldn't compare it with mitochondria necessarily, but so each domain, let's say, is a node, and
yes,
it's a reinforcement learning system in itself.
yes,
But then it's all connected into a network within the genome,
that's
right?
correct.
And then it, yes, adapts and it learns at the same time. But
Yeah,
in part, the reason for it, and it's done through individual domains as well, because again, each individual domain is a reinforcement learning system.
yes,
But then the whole network adapts as well. And then probably an important reason for that is to process inputs from other cells, forming a bigger whole.
right. And we had Daniel Phillips present with Dennis Noble about um, the um, um, the vesicles carrying RNA, which
Hmm.
actually get transmitted Lamarckianly, I'll say, because I prefer that term to epigenetics. I think it's much more descriptive, back to the germline.
Hmm.
Okay. And so if we have I've never seen a
Hmm.
cell not conserve a mechanism. And if it's used in that way, why wouldn't it be used to hold memory in some fashion for the nodes themselves?
Hmm.
And and it also fits with the recursivity of a holon. These things tend to repeat at larger and smaller um, um,
Right. No,
intervals.
I think it's a really interesting. Yeah, I would like to think about this one. It's a very interesting consideration. And also, I don't know if it's really related or not, but I mean, these domains are fractal. The whole
Yeah,
idea of
it's a
fractal
fractal
is
process.
the,
Yep.
yes, it's
And your fractality is beautiful because it's a chaos fractality. I think we need to and this is another perfect example I was going to mention. People stop looking at life. I think most of us have adopted the idea of constraint closure at this time, and most of us stop looking at life when we don't see an obvious metabolic cost that involves, a mitochondria or the like in a primitive organism, right? What if the metabolic cost is, in this case, so you might look at your nodes and say, well, they don't have a mitochondria, they're not maintaining themselves. They don't have enzymes that they're actually creating. What if part of that metabolic cost is the circuitry that's held and managed through the capacitance at the boundary?
mhm,
We've just determined that there's membrane, which means, and you've told me that you said these are electrical entities. So what if that is part of the metabolic cost that's being maintained? And we shouldn't look at everything in terms of, and what if the other part? This is something I've been looking at a lot in terms of rosin. A lot of folks are saying that rosin is just lipstick on thermodynamics. Well, perhaps to some degree, what that means is that we should be looking at energy costs and energy transduction and sharing and generation at other than the process that comes through ATP the ATP process, the Krebs cycle and so forth.
mhm.
Does that make sense?
That does make sense. Another thing that I was just thinking about, again, maybe it's relevant, maybe not. So and it's conjecture at this point too. How did this intronic and extragenic elements that ended up forming the main volumes. How did they ended up in the cell? And well, I don't think anybody knows for sure, but a lot of them are things like line of one elements, for example. And so a lot of them, presumably, at least what most people believe right now, they were delivered to our genome virally
Virally, that's
by
correct.
by,
So
by
is a
viruses.
virus, so how is a virus a holon?
Right.
That's another question. Or is it a pseudoholon? I do believe that this is a set of fractals that life is throwing up over and over, and
Hmm.
perhaps they're fractals that do actually aren't infinitely, regressed. Perhaps there's a place at which
Right.
they are a pseudoholon.
Hmm.
Do you know what I'm saying?
Yeah, yeah, I know what you're saying. Yes. Very interesting. Yeah.
Well, I could talk about this all day. Maybe Charlie will set a time when we can
that
get
would be
back in
great.
it.
Yes.
Or we could, I'd love to speak with you privately, Vadim. It's your work. At first I thought, what is this going to be? And then all of a sudden it was like, holons everywhere. This is so fascinating. And you've really mapped out a lot of it.
They are everywhere. Yes.
Yeah.
That's great. Awesome. Henry?
Okay, so during the listening, I mean the previous two discuss. I mean, one is what's the difference between bacterial and between eukaryote. So for a long time, we're actually thinking, so if bacteria is so successful, why evolution will bring the new category of life, right?
Hmm.
So that
Hmm.
is very interesting thinking. And then we realized that the driving force, or maybe the core mechanism of evolution, is not about the success of different species, or even not the biological system at all, but rather is a mechanism to increase the information complexity. So that's our
Hmm.
new theory now. So because just by coincidence, the life is a very effective platform. That's all. So if we think in this way, then we look for bacteria, we're looking for eukaryotes, we're looking for consciousness, everything, right? Look at AI even right now. So it's the same idea of pass and create and preserve and the usage of this information. So now all of a sudden we say, okay, this actually answer many of the questions, including congregation and why the congregation media and evolution is so important, because that is a platform which can fulfill this goal. So for example, bacteria actually, if how the people classify bacteria, they use ribosomal RNA, how the similarity about 60, 80, oh, this is a species. But in fact, they don't have the identical species. Why? Because they don't have the mechanism for the inheritance. They only have the DNA, molecule ATGC. It's very rough. Not like the, eukaryotic system, they have sex, they have DNA pair, they have environment, everything is about the information constraint. So we actually have been all around for a long time. We're always thinking evolution, innovation, too much innovation, but evolution is all about conservation, what you just created. So the bacteria constantly create a new one, but they constantly come back. So after 3.5 billion years, they go to nowhere because they don't have such a mechanism to preserve what they just created, right? So for example, bacteria under stress, they also form the huge bacteria, but they immediately come back and they goes to nowhere. But for embryonic, for the cancer, when they form that one, they immediately become new system. So every time they go there, become new system. So they continue going because they have the inheritance to back them up, right? So we're looking for all the animal, the development process constraint, all the physiological condition constraint because everything is constrained. Why we cannot say that? Because we love evolution, we love novel, but we forgot the two faces of same coin, right? So bacteria is the same thing. They create every day. Actually, the clone concept is wrong. If you sequence each E. coli, they're all different every time. They're not clone. They're completely chaotic process. But however, in our mind, we always think it's clone, right? So that's the problem. So when we found that the function of sexual reproduction is constrained rather than provide diversity, we constantly realize all the evolution theory had fundamentally flawed because of this, right? So that actually is very important. So therefore, my question is, because people right now say, oh, we do not need to think about inheritance because say if we put the cell into different environments that have different feature. So what importance is the inheritance? But people forgot what we are doing. All of this is artificial experiment. We take the ecology, take the condition away. We said, oh, they can pass, but pass to where? Right? So that's the problem. That's the fundamentally problem. Therefore, we realized no matter how beautiful, you have epigenetic, you have consciousness, everything, you have to pass to next generation. By what? By inheritance. So people actually stuck into ridiculous, man sitting there to argue whether not important, not important. Of course, it's important. Of course, for example, we can do embryonic manipulation, get lots of stuff, but this knowledge is not about the biological in nature. It's biological in our hands. We use the technology, we can teach them up, and we artificially build the constraint. We actually fool ourselves in a way, we did not realize, right? So that's actually is very important. So therefore, we thinking karyotype level genome is the code of the codes, because epigenetic and methylation conscious, they have lots of codes. Every time the biological system just build up, move forward. Therefore, they have phenomena complex, speed up the complexity. So the bacteria are too simple. They don't have the way to do that. We actually showed an experiment in the cancer cell under massive drug therapy. The chromosome instantly shuffling. Every time shuffling, they become new high order of system. But the bacteria do the same thing, but it's always the lower level systems. So because evolution is not like Darwin's claim, small accumulation, you accumulate become big. No, never happened. The bacteria is the example. They always change, but because they don't have constraint, they're always there, right? So therefore, the mechanistically, they have microevolution miracle in bacteria, but never have macroevolution miracle. So that's what actually I really thinking this. And secondly, I use your work. We should more, push the idea that the coding is not the products, is the process. And then people can actually reconcile people's argument. I mean, so much argument is because we're thinking coding is linear, but it's not. But the coding process must be there. Otherwise, there's no evolutionary products. So
I would say that I would love us to write a review article on this at some point, because a lot of what you said is, a lot of the ideas, what you just mentioned, are very much along the lines of what internally we have been discussing in my group. So and I think in part also goes back to the, question of the selfish gene, right? Which fundamentally is the question, what is life, right? Or what's the purpose of life, right? Is it to pass a gene? And if that is the goal, then all bacteria are doing a pretty good job at it. Well, actually, nobody does a good job at it, because you always lose, in a sexual reproduction, you're losing 50% of your gene. But if anything, bacteria are doing really well in this regard, and that should be the end of it, right? But I think there's a fundamental question, what life actually is. And I can tell you my kind of personal take. If I need to answer this question, I probably would not quote what Wikipedia says. Like there's what, 6, 7 criteria for life, like heavy metabolism, life, death, procreation, this and that, adaptation. Uh, I think there is and it's my personal opinion. I may change it, a year later. But I think life is more about processing information. And why is that? Because the key is to being able to consistently lower your own entropy, organismal entropy, at the expense of the entropy of the, well, outside world, right? But the only real way that we know how to do it is by, right? So if a system can process information, it can lower its entropy, right? Otherwise, it will be a refrigerator. Somebody else creates it and just keeps the door, and the entropy will eventually go up as well, right? So processing information, doing computations that seems to be the critical part. So now, do bacteria do computations? Of course they do, right? They have GRN, they have some adaptation. Of course they do. They have chemical networks, they do computations. But these are fairly simple computations. Eukaryotic cells in us, we are doing much more complex computations. So there is a there is a drive to higher levels of computations, I think. The only way to get there is to invest in individual cells in individual being able to do more complex computations. How do you do that? Well, you cannot do this at the population level. The whole idea of the population is that an individual doesn't matter. Individual bacteria can die. The population of the whole species will propagate. But if you begin to make individual cell or individual more and more complex, you can get there. But the essence is not that it's the propagation of a gene that matters in this case. And I think I agree with you. It's not the propagation of species per se. It's the ability of the entire system, not just an individual cell, not just an individual organism, but the entire system, entire ecosystem, if you will, to be more computationally efficient and do more complex computations. Uh, the same thing that we know very well from computer science is that a single computer can do so much. In a supercomputer with consists of multiple nodes, cluster of computers can do much more complex, more efficient computations. And that I think I'm probably more comfortable with this concept personally, that it's the computational complexity of the entire life that matters in the end. And from this perspective, I think the message is a little more altruistic than selfish gene. It's not about propagating my gene. I don't matter. My gene doesn't matter. I only matter as a part of the computational life system as a whole.
Yeah, that's exactly what the cancer did. Every time they pass out themselves, they just pass the information and let others to carry on the torch. So anyway, yeah, thanks.
That was beautiful. Giovanna?
Hi. So just in relation to one of the previous questions, I guess. So can we claim that the genome itself exhibits the capacity to learn? So does it as a particular pattern can itself exhibit some kind of cognition or agency or again this kind of reinforcement learning, or is it the organism that learns and then that the kind of the consequence of that learning gets propagated into the genome, which is in the end kind of like a mechanical plate onto which these changes or epigenetic changes and the processing of information is kind of taken down, which again gets continuously reinterpreted by the organism, right? So again, can that pattern even have some kind of causal power if, and can it exist as such, if there wasn't for an organism to hold it, to interpret it continuously, to preserve it, right?
So I think it's a great question. I think the answer probably will depend on what we define as computations, right? But if we define computations as the ability of a system to take inputs and generate outputs with the uh, and project the environment, external environment, meaning the matrix of stimuli on itself, then I think clearly the genome itself is a computational system. I'm not arguing that I'm clearly not arguing that it's a self-aware system. Uh, that's but it is a computational system, and from this perspective, it it's a quite intelligent system at that. It has its own intelligence. Uh, it can change, it can adapt, it can change its own weights. In some ways, it's a smarter system than many of the AI networks that we've been creating with fixed weights and fixed learning structures. Um, I mean, clearly it doesn't exist in isolation, but I do find it kind of a little eerie in the sense that every cell in my body, inside its nucleus, has its own little computer, its own little intelligent, not just computational, but intelligent system. I think it satisfies the rules for what we would call intelligence. And it has its own agency, not to be confused with self-awareness. I'm not saying it's self-aware.
Thanks.
Great. That was great. I think that might be a great place to leave it, guys. What a beautiful conversation. Great job.
I learn a lot, and I really enjoy this, and thank you.
Yeah. Yeah,
They're really good ideas.
we really enjoyed having you, Vadim, and I hope you'll come back and join as a participant in future
I
meetings.
I would love to. I would love to be back.
Yeah, you'd be very welcome. Great. Alicia says, thanks. Terrific. In the chat.
This is great.
Awesome.
Thank you so much, Vadim.
Yeah.
Thank you. Thank you for your ideas.
Yeah. Great. And we'll share email addresses and I think your academic appointment gives you that email that's easily searchable too. So um but um but anyway, it's
All
been a
right.
good one. And
Thank you.
we'll all be back about 3 weeks until Stuart Kaufman's next session. So we'll look forward to that. Thanks so much, guys.
Thank you.
Bye bye.